first one is Professor Lewis Vitetta, and it is indeed a pleasure to introduce Lewis. He's Director of Medical Research at Medlab Clinical and Professor with the University of Sydney in Sydney Medical School. Between the years of 2007 to 2013, he was Director and Professor of the Centre for Integrative Clinical and Molecular Science at the University of Queensland School of Medicine. Lewis has extensive research interest in the microbiome, nutrition, probiotics and immune function area, as well as research on cannabis medicines. Over to you, Lewis.
Thank you, Christine. It's always good to come to Melbourne. I always like coming to Melbourne. I grew up in Melbourne, so it's a really nice place to come here. It's better than Sydney. And we didn't bring any cannabis samples, even though Sydney's loaded with them. Okay, look, when they asked me to do this and to talk about the intestinal microbiome and pain, there is a significant amount of evidence, but in the time allocated, I don't think I can do everything, but I'll give you some very interesting examples as to how the intestinal microbiome is actually significantly associated with the development of a lot of diseases that exhibit chronic pain. Okay, the bacteria are ubiquitous on this planet. They're at the bottom of the Antarctic ice. They're on the land, in the soil, on plants. Definitely, they're on all types of animals. They're in salt water. They're in freshwater streams. They're also present in volcanic pools, such as you would find in Yellowstone National Park in the United States. So we need to really understand that they are everywhere. And that when you look at the evolution of species on this planet, we can actually see that bacteria are very much allocated everywhere, and that eukaryotes, which is what we're made up of, actually we came from a bacterium. If you really look essentially as to how eukaryotic cells were actually formed, there was a particular type of alpha proteobacteria that then changed its shape and became a eukaryote, and then we went from here to something that looks like this. So it's very interesting to actually see this, because when we then have a look at ecological maps associated with the bacteria that live on and within humans, it looks something like this. And where we have an established human microbiome with the major phyla being these, Firmicutes and Bacteroidetes, which make up about 95–96% of the bacteria, let's say in the intestines. Then we have a couple of others, Actinobacteria, Proteobacteria, Verrucomicrobia, which is a very interesting phyla. It's composed of less than 1% of all the bacteria, but it has a very specific member, Akkermansia mucinifera, which is a very important microbe in the gut to turn over mucus. And that's very, very important, because we need to be turning the mucus over on a daily basis. And the bacteria under this phyla tends to do that very, very significantly, because when we don't turn mucus over in the gut, we put ourselves at risk for developing a dysbiotic gut or leaky gut, that, as you will see in a minute, that then puts us increasing risk for developing metabolic disorders such as type 2 diabetes, chronic kidney disease, rheumatoid arthritis, osteoarthritis, and certain other mood disorders. Now, when we were starting to look at this, we actually found that there was an overarching influence of the gut microbiome on end organ function, and that the gut was at the centre of developing adverse reactions associated with a whole number of organs, and that the bacteria in the intestines was actually the major contributor, the major trigger to adverse outcomes associated with fatty liver, pancreatic dysfunction, mood disorders of any type, lung functionality, skin problems such as eczema, and respiratory problems such as asthma. So in looking at all of this and understanding that the bacteria are primordial in I guess, moving forward, adverse pro-inflammatory states that are then connecting to various end organ functions, we really wanted to know, well, where is all the pain in this? So if you look at the types of pain that exist in clinical practice, we have acute pain, we've got chronic pain, And what I wanted to do is just to actually very much look at chronic pain tonight, because there's not enough time to look at everything. And when we're looking at chronic pain, it's associated with gut microbiota adverse shifts. For chronic prostatitis, chronic pelvic pain, visceral pain, migraine, fibromyalgia, arthritis, whether it be osteoarthritis or rheumatoid arthritis. And within that realm, the impact of chronic pain can have significant, per employee costs over a particular year with respect to anything from cancer pain, any form of cancer pain, any form of arthritis pain, any type of back, neck disorders, neuropathic pain, and with respect to direct and indirect costs, the bacteria are actually determining the cost. And when you look at what kind, and this is a very complicated figure, and I apologize for that, we have a diagrammatic representation of what the intestinal epithelia looks like. It's a single layer of cells that keeps at bay 10 to the 14 cells per milliliter of fluid in the large bowel from the mucosal system that is associated with neuroendocrine immunological activity that controls a lot of the functionality with respect to end organ physiology in order to maintain some sort of an equilibrium. So we're basically set up to maintain an equilibrium in the gut. And when the equilibrium is off, the immune system is off, bacterial products are off, the vagus nerve gets upset and sends negative signals to the brain. And in terms of the intestinal epithelia, it becomes leaky. And leaky means that things from the lumen move into the mucosa, triggering pro-inflammatory activity that's out of control. And in terms of the dysbiosis, disrupted gut homeostasis, there are a number of certain factors, like, for example, dietary practices that are not very prudent that lead to obesity, stressful situations. And if you've heard me talk to you before about stress, everybody's got stress. When I did a PhD at the University of Melbourne and I did 613 postmortems with Avni Sali, well, he looked on and I did the work. None of those people looked stressed to me. They got something else. Lack of physical activity is a problem, and poor sleep is a problem as well. And we've published a number of areas in this area to look at intestinal dysbiosis risk. And here's what you see: the altered gut microbiota, impaired nutrient absorption, tight junction proteins tend to break down and things leak into this area of the mucosa where you now have an immune activation and gut barrier dysfunction. And what we then see is that in terms of diet, high-fat diet, high-sugar diet, high alcohol consumption, the lack of use of functional foods, biological stress that disrupts circadian rhythm. You've got a number of conditions that actually become high risk when the intestinal permeability is increased and the microbiota is now in an unstable environment that then leads to increasing risk for developing a chronic disease. And it's important because the etiology of leaky intestines shows that it's associated with nutrient malabsorption, increase in risk for autoimmunity in those that have got a genetic susceptible risk for, food intolerances, blood-brain barrier breaches of toxins in the bloodstream that then cause an additional problem with respect to mood disorders, and systemic inflammation associated with joints, like, for example, osteoarthritis, rheumatoid arthritis. And that is, I think, problematic because this is a general disturbance of the gut microbiota equilibrium that's due to an imbalance of the bacterial cohort. And the bacteria have often been claimed to have 10 times more bacteria than human cells. That's not true. It's more about 1-to-1. And the changes in their functional composition and the metabolic activities and changes that they produce in their local distribution has now become very, very significant in putting us at risk for developing a type of adverse condition with respect to increasing the risk for a chronic disease, because it's all about risk. It's about increasing or decreasing the risk for developing a chronic disease. And the loss of beneficial organisms is very, very important. 1 round or 2 rounds of antibiotics in a newborn in the first 6 months of life can actually significantly reduce keystone species that you and I then can't recover. And that individual is now at risk for developing a chronic disease in later life. The overgrowth of pathogens. Pathogens are those bacteria that don't tolerate the host. And often when I talk about bacteria, people say, oh, the good and the bad bacteria. There's no such thing. There's just bacteria. The ones that tolerate the host and the ones that don't. If you were to get a probiotic that everybody says this is a good bacterium and you inject that into the bloodstream, you're going to get a very nice surprise. They don't belong there. They belong in the gut. So we need to really understand what we're talking about in terms of pathogens. And the loss of overall diversity and possible abundance is also a very significant risk for developing chronic diseases. And aging is a disease. And when you look at supercentenarians in some of the work that we have done compared to those studies that have come from the Mediterranean area, Italy and France, what we find is that supercentenarians have a very diverse and abundant intestinal microbiome compared to those people that succumb around 70 and 80 years of age. And one of the main bacterial phyla, or I guess families, that is in deficit as we age is the bifidobacteria. And bifidobacteria are very, very important because they handshake with the mucosal immune system every minute of our lives. And when we don't have those bacteria, the mucosal immune system in the gut tends to go into some sort of demented state. And when you have a look at, and if you're in clinical medicine and you have a look at what goes on with the elderly who don't have chronic diseases, what do they mostly succumb from? Some sort of chest infection that becomes pneumonia and they succumb. And this is an altered gut mucosal immune system that has been decimated because they don't have overall diversity and abundance. They have actually lost that. So it's important to look at the bacteria. The two major phyla, Firmicutes and Bacteroides ratio, when these ratios are shown to be of significant relevance in signaling gut microbial status. Increased Firmicutes to decreased Bacteroides is a signature of intestinal dysbiosis, and this is leading to pain. Relative changes in the abundance of these two phyla, certainly these two dominant phyla, are seen in obesity, in chronic stress, in depression, in irritable bowel syndrome, and in a number of other chronic diseases, such as chronic kidney disease and type 2 diabetes. So the intestinal dysbiosis risk, as we've seen in this diagrammatic representation, tends to lead to systemic inflammation. And systemic inflammation then connects the gut, the bacteria, a mucosal immune system that is out of equilibrium with changes in adipose tissue inflammatory responses, liver problems such as non-alcoholic fatty liver disease and the deposition of extra fat, and skeletal muscle problems. And skeletal muscle problems, this is really important because the gut bacteria also determine the loss of skeletal muscle mass. And that, for me and our research, is the #1 marker of aging. The quicker you and I get skeletal muscle mass loss, the quicker we have a foot in the grave. And so we want to be keeping as much skeletal muscle mass that we can in terms of keeping the bacteria happy, abundant, and diverse, and doing some extra bits and pieces such as some 20 minutes or so of weight-bearing exercises in order to actually keep our muscle mass. And that is important because the skeletal muscle is loaded with mitochondria. And mitochondria are the energy organelles of the cell, producing a lot of energy. And mitochondria have their own DNA, and they turn over. Muscle cells turn over once every 30 days. What's very, very important is that a piece of that DNA in mitochondria is a taxonomic marker for bacteria. So the bacteria actually gave rise to the mitochondria. So you can see there's an interesting connection between what's going on in the gut and maintaining skeletal muscle mass so that we age slowly. I don't have any drugs for that yet. The microbiome regulation of visceral pain is rather complicated, and I apologise for this. Anybody that remembers any of this is crazy, so you shouldn't be remembering any of this stuff. But just to know that there are the bacteria producing short-chain fatty acids, which are very, very important, and our discovery is that butyric acid, butyrate, a short-chain fatty acid that is produced in abundance by when you consume prebiotics or probiotics by the intestinal microbiome, tends to actually maintain the mucosal immune system in some sort of an equilibrium, and that is a benefit for you and I every day of the week. And then we've got other types of inflammatory cytokines that are actually controlled, and we actually down-regulate certain pain pathways by just producing short-chain fatty acids that maintain dysbiosis at bay in the gut. Okay. Now, there are a number of medications that have been published over the last decade or so that can disrupt the intestinal microbiome. Certainly, antibiotic drugs do this. A number of psychotropics, proton pump inhibitors that tend to... Proton pump inhibitors are those drugs that control acid production in the stomach, and they reduce acid production in the stomach in those people that have got some sort of reflux esophagitis problem. And in terms of doing that, it changes the microbiome in the small bowel from what should be there to what is in the mouth. And we now have a change in the microbiome in the small bowel that causes a dysbiosis up in the small bowel that then puts people at risk for developing certain things such as small intestinal bacterial overgrowth, which is a problem that many patients do present with in terms of proton pump inhibitors. But there's also, with proton pump inhibitors, an increase in risk in developing some very serious infections such as those associated with Clostridium difficile, because they become more susceptible to that. And also extensive impact on non-antibiotic drugs on human gut bacteria, which really happens on a regular basis in most clinical practices of the drugs that people are being prescribed. This is just a diagrammatic representation of what's going on in the gut with respect to the intestinal villi, and this is just, I guess, an expanded view of these. Just to show you what really goes on here. The bacteria in the lumen tend to continuously shake hands with the intestinal epithelia so that the mucosal immune system down here actually understands what's going on with respect to a pro or an anti-inflammatory signal. We live in that controlled state. We're a little bit inflamed, we're a little bit non-inflamed. When we are super inflamed, we have a problem. We have a risk for disease, such as Crohn's disease, irritable bowel syndrome, ulcerative colitis in the large bowel. And when we're not responsive, we have a necrotic gut, and that's a problem as well. So the two extremes are a problem, and we need to be equilibrated. So in terms of the medications in the intestinal microbiome, I think one of the best examples is what morphine does to the microbial cohort in the gut and inducing dysbiosis. It can disrupt cholesterol bile acid metabolism, which are very, very important for actually maintaining the equilibrium of the bacterial cohort in the gut. The bile acids are very, very important. It can also disrupt the intestinal barrier significantly by actually altering the small and the large bowel epithelia. The epithelia are joined together by proteins, zollulin 1 and zollulin 2, and occludin 1 and occludin 2. This is these funny names for these proteins, and they're held together so that I guess you would say that the barrier is not significantly leaky, but things like morphine can actually disrupt that leakiness and make it even more leaky. And you then end up promoting a sustained inflammatory response in the host that then needs some sort of an intervention. These adverse shifts have been associated with immunity problems, inflammatory problems, certain motility problems in the gut, and also neurological changes that are the hallmark characteristics of functional gastrointestinal disorders. Because when we look at the work that we do with depressed patients, what we find is that they are patients with major depression all have a significant dysbiotic gut that is disrupted, and without recovering of that dysbiosis, some of these patients then become refractory to antidepressive drugs. So they're not even responding to the drugs until you can actually fix the dysbiotic effect. Now, what do we know all this? Why is this really important? I was working on bacteria before the Human Microbiome Project began looking at things to do with the human microbiome, and that began around the early 90s. And what's important from that study was that the National Institute of Health then showed that in key experiments with germ-free mice, showed that if these animals did not have any bacteria, they didn't come in contact with any bacteria, all of these processes suffered. And when these animals then were freed into, into the wild, they succumbed very, very quickly due to some very horrendous infections, because the bacteria were not there to shake hands with the immune system of the animal to actually get them to acquire immunological tolerance. And that is what newborn babies try and do for the first 3 years of life, is to achieve immunological tolerance. And it's the bacteria that actually help the baby do that. And that's very, very important, because when we look at animals that are raised in a sterile environment, they have an exaggerated stress response to reduced perception of pain. So when the bacteria are not there, they don't respond very, very well to pain, and they have very, very different inflammatory stimuli. And this is important because antibiotic-mediated depletion of intestinal microbiome, when you then do this synthetically, it decreases visceral pain-related response in these experimentally naive mice and rats. And that then tends to put an early life exposure to antibiotics, predisposing the animals to visceral hypersensitivity in adulthood. So what happens to these animals early on is then imprinted and taken on into adulthood. And it's important to understand this, because the fecal matter from viscerally hypersensitive IBS patients, when transplanted to germ-free mice or rats, end up actually responding with a distension of the colon that is very much enhanced in these animals. So if we get a fecal sample from a patient that has irritable bowel syndrome and we transplant it to one of these germ-free animals, they end up responding with an increased colonic problem and pain. So we can see that, again, the bacteria are actually showing us that they are actually dictating the way, I guess, that the microbiome in the intestines actually controls visceral pain in a number of different conditions. Can probiotics fix this? Probiotics are not a panacea to fix all chronic diseases, but there is accumulating empirical evidence that supports a role for probiotics which positively influence the intestinal microbiome in the treatment of visceral sensitivity. And this is important because provided the vagus nerve integrity is maintained, this can actually be significantly influenced. And in terms of an example such as fibromyalgia, which the symptoms are associated with fatigue and impaired sleep and cognitive difficulties, these fibromyalgia, if you don't know what this is a musculoskeletal problem, very much, I think, of unknown etiology. It's a disorder that's characterised by widespread musculoskeletal pain accompanied by fatigue and sleep and cognitive difficulties. And the research controlled for factors. When you do work on fibromyalgia, they control for diet and they control for genetics and physical activity and the age and so on, in order to be able to assess what really is going on in the muscle and relate that to the intestinal microbiome. And it's important because a recent study published in Pain showed that in patients with fibromyalgia, they had an altered microbiome composition in individuals with fibromyalgia as compared to those that were controls of the same age and same gender. The particular research showed that in a cohort of 156 individuals in the Montreal area in Canada, 77 who were diagnosed with fibromyalgia, approximately 20 different species of bacteria were found in either gender or that were either greater or lesser quantities in the microbiome of the participants suffering from the disease as compared to the control group. So you can see, as you find that there is a chronic disease problem with significant pain associated with skeletal muscle, there's a significant shift in the intestinal microbiome cohort. And if I was to just show you this, and I apologise, this is again, you don't have to remember all of this, but what you see in the two individuals, that there is a significant change in a number of bacteria that is associated with fibromyalgia versus these that are not associated with fibromyalgia. And Faecalibacterium prausnitzii is a very, very nice bacterium to have a lot of. And when you don't have it, there's a lot of problems associated with other bacteria that come along and take over the niches of these bacteria, causing a disruption in the intestinal microbiome cohort that then leads to increasing risk for developing fibromyalgia in patients that are susceptible for this. So it's a very interesting study. It's probably the first of its kind that's actually showed that there is a change in the intestinal microbiome associated with fibromyalgia. In other intestinal problems such as rheumatoid arthritis, which looks like this in the joints, there's also been studies that actually show that there is a decrease in intestinal microbial diversity. Again, the same story. More pain, more changes in the intestinal microbial cohort that's associated with reduced diversity, reduced abundance in patients with rheumatoid arthritis. Changes in intestinal bacterial profiles correlated with increases in inflammatory profiles. So it's very, very simple to really understand. From a clinical perspective, it's very easy. From a scientific, wanting to find out what really is going on here, it's a bit more difficult. But there is a loss of diversity and abundance that is very much associated with those bacterial cells that actually support the host, that tolerate the host. And there is a significant increase in pathobionts, these bacteria that can become pathogenic under certain conditions. So what we have definitive connections between the intestinal microbiome, adopting prudent dietary practices that promote a balanced and diverse level of gut bacteria is very much advised. And I would say that foods that include in the diet, fermented foods encourage the intestinal microbiome to be diverse and to be rich. Dairy of the kind of low fat-free milk, yogurt, and the like are also very, very good. Probiotics formulated by research. You just can't go and get any probiotic and think you're going to get something. You better be very careful with that. Because I do a lot of research in this area, and there's a lot of probiotics on the market that have nothing in them. So be careful with your money. That's not a commercial for Medlab, all right? It's not about Medlab. High fiber foods, fresh fruit and veggies and whole grains is also very, very good. And anti-inflammatory foods such as beans, cold water fish, walnuts, flaxseed, soybean, all very, very good in order to maintain the intestinal microbiome variable, rich and abundant. And one last bit that I want to talk about is altered gut microbiota and the endocannabinoid system. The endocannabinoid system has been associated with cannabis products that my colleague here will talk to you about. And the data from this murine study suggests that there is a link between vitamin D deficiency with changes in the gut bacterial composition, and there's altered actually perception of the pain. And I wish we knew more about the molecular mechanisms, but I think that some of them involve the endocannabinoid system and the related mediated signaling systems. This is a lot of biochemistry mumbo-jumbo stuff that you don't really need to know about. But what he's saying to me is that the bacteria are producing certain metabolites in the gut that talks to the endocannabinoid system, that system that's associated with cannabis receptors 1 and 2, that will then re-establish some sort of, I would say, signaling equilibrium in the neuroendocrine immunological mucosa. That's a big mouthful, all right? But that's where it's at, because the problems for me in the gut have always been in the mucosa and not in the lumen. The lumen is just the area where the bacteria are altering their abundance and their diversity, but it's that reaction in the mucosa that then leads to some sort of triggering pro-inflammatory activity that then is associated with pain. And what is interesting about this study is that vitamin D. And vitamin D, I think, is really not a vitamin, it's actually a hormone. And every somatic tissue is associated with vitamin D. And where is the receptor of vitamin D? It's on the outer membrane of the mitochondria. Again, another relationship of mitochondria, an important nutrient such as vitamin D, and also the association with neuroendocrine immunological dysfunction and the triggering of pain. So it's interesting to see all of these things. This is what we do at Medlab. In my lab, I'm very proud to have some of the people that I work with. I've got 5 PhD graduates and 2 PhD students. We look at a number of problems that are associated with the health system today, from pain management, which involves our cannabis medicines, to depression, obesity, metabolic disorder, chronic kidney disease, and the age and skeletal muscle. This is a cannabis. We made friends with New South Wales Health, and they made friends with the police, and they used to bring us cannabis, which was really good. But I'll tell you something. In 1 year, we had 5 kilos of cannabis, about $500,000 worth on the street value, and 3 of those came from 3 areas outside Sydney that had nothing in them. And people were paying up to about $50, $60 for a couple of grams for that stuff. So you've got to be very mindful that if you're going to look at cannabis for managing pain, that you need to be really looking at what it is that you're getting, and don't buy any street stuff. All right? Listen to this lady here afterwards. She'll tell you exactly what you've got to do. What we do at Medlab is we nanosell things. We make fat-soluble compounds into water-soluble compounds. This is beta-carotene. You can see it's not mixing with the water. And then when we nanocell it, it becomes water-soluble. And that's what we do with our cannabis medicines, also with vitamin D, with B12, with CoQ10, with insulin, with amoxicillin, a whole bunch of molecules. And what we think we do is that this thing just crosses the oral mucosa or buccal mucosa. This is the inside of the cheek. Very, very quick traverse into this into the systemic circulation via the facial lymphatics. This is a very rich vascular area, so I think that we do something very well with this. It's another way of looking at things in order to bypass what's going on in the gut, especially with cannabis medicines, because look at the activity that's going on here. You know, there's all these activity, commensal microbiota and their chemicals, and probiotic bacteria, what they do, and then you've got secretions of active molecules, what's going on in the mucosa between pro-inflammatory and anti-inflammatory activity, and the macrophages, and really what's the modulation of immunity. So the central role of the gut is to control all of this. And if you're interested in cannabis medicines, in controlling pain, well, you need to be very careful as to what it is that you are ingesting or applying or administering in terms of managing your pain. The central role of the gut is to actually maintain, in early life, to have a microbiota development that triggers immunological tolerance for that newborn. That will happen over a 3-year period. Once that 3-year period has passed, that newborn or that child now has an adult microbiota profile. It's important that they have immunological function that is maintained in equilibrium. Digestion and absorption is important for metabolism, for organs such as the liver. Defense, immunological defense is important. Tolerance is associated with good nutrition. Satiety, mood and pain, gut-brain crosstalk between what's going on here and the brain is also very, very important because of the vagus nerve and because of the circulation. The vagus nerve is the only cranial nerve that innervates the gastrointestinal tract, whereby 80% of the fibers go from the gut to the brain, and only 20% of the fibers go from the brain to the gut. And the vagus nerve actually senses the microbiota tone, whether it's dysbiotic or in equilibrium. And that's important because it will actually maintain a dysbiosis at bay. I think I've hit all the points that I wanted to. I always have to mention, these are the people that I work with. Nothing that happens that I do works by myself. Tamara will tell you a hell of a lot about cannabis. She's got some very nice slides to share with you. I thank you. I thank them.